Hello, this is a short video about the neural foundations of dynamic field theory, part of my lecture course. So in an earlier lecture, I talked about the brain just in very general, and I had this picture from a textbook that's cited here, in which is a very rough assignment of function to different parts of the brain. For instance, here the back is roughly the visual cortex, is the motor cortex, frontal cortex, and there can be many other function assignments like that. See here the visual information from the eye goes to the visual cortex, and the activity in the motor cortex goes to the motor output. So that's a typical way of thinking about the brain. And how does information like that come about? How do we assign functions to particular subsets of the brain? Well, the two fundamental ways that this is done, and I'll talk about the first one, which is roughly described by the keywords of coding, decoding, and it's part of what is called cognitive neuroscience. And that's the idea that you're observing a brain with different techniques while you vary something that's outside the brain. That could be a stimulus, it could be the motor task, could be a cognitive task, and so on. And so as you vary that outer stimulus, you look at what happens to the activation. I'll go deeper into that in the lecture. There's also the reverse approach where you vary the neural substrate. You can do that by lesioning the brain, deactivating certain regions. There's now very advanced work where you use optogenetics, and you can sort of switch on and off networks, and then you observe what happens to behavior, to competence. And that's classically called neuropsychology, and there are much more advanced versions of that. And I'll only briefly refer to lesioning in one little spot here in the lecture. Now, in this coding decoding picture, the fundamental idea is what is called the tuning curve. Is you observe neural activity. For instance, if you really record from individual neurons, you can look at the spike rate as a measure of the activity. As you vary some outer dimensions, so here, for instance, as you vary a stimulus parameter, in the chapter 3 of the book that I'll make available, is reviewing this in more detail. So here's a classical example from Hubel and Wiesel's 62 paper. Hubel and Wiesel got the Nobel Prize ultimately for finding out the functional architecture of the visual cortex, and their work, which was related to it wasn't not in a vacuum, but what is now associated with their names is sort of was set that paradigm, the paradigm of looking at coding as a way to characterize functional properties of the brain. So they looked at visual area V1 or area 17 in the cat. This is monkey work in which they recorded from neurons the spiking rates. These would be spikes here. As they were shining light onto the retina of this anesthetized animal, and they found that light shone into an area that is outlined here by the dashed line would elicit spikes, while outside that area there wouldn't be any spikes. This area is then called the receptive field of the neuron recorded. And they also found that when a bar of light is moved across the receptive field in a particular direction, like this direction, an elongated bar, then you get a lot more firing than if it is moved in some other direction, and that also implies different orientation of the bar. And so if you look at the firing rate of the neuron as a function of that orientation, that happens to be also the movement direction here, then you get a very nice tuning curve like that, where maximal firing rates here in this anesthetized preparation, about 50 hertz, arise at the optimal angle for that particular cell. And then very typically you would characterize the cell by that optimal stimulus that drives the cell optimally. So that will be a tuning curve, right? Relating something inside the brain, this here, the firing rate of that neuron, to something outside the brain. Analogous in the motor system, this is from Jacobus's lab. I'll come back to that later. You have a monkey make different movements, different tasks, moving a handle in different spatial directions, and then you look at the neurons in the primary motor cortex, for instance, these little spikes here in different trials, and find that they vary their activity as the monkey makes or prepares that movement. And you see that the neuron fires more for these kind of movement directions, less for these kind of movement directions. And you could similarly look at the direction in which the movement takes place how that predicts the firing rate of the neuron across these different movements. And it's also a tuning curve. So actually cosine, very broad kind of tuning curve. So this is the classical concept of coding. Actually, decoding is sort of the optimized version of that, where you have a pattern of neural activity that you observed while you were at the stimuli, and then you're assessing to which extent you can predict what stimulus or movement condition or task condition was the case from the neural data. And the extent to which you can do that tells you how strongly the neural activity is associated or predicted by the task. And now, if you do that for a lot of neurons and look systematically across the cortical surface, you can find out about how many different neurons, how their dependence on the stimulus parameter, their tuning curves varies. So in the simplest case, for instance, you could have a spatially structured stimulus and then record across the cortical surface, and you will often find that the spatial structure of activity across the cortical surface reflects the spatial structure of stimulus, a principle called topography, that is neighboring cells represent neighboring things here, neighboring things on the cortical surface. If you look at other parameters, like for instance, which eye primarily drives neurons, and you find that there's also some regularity, that there are certain stripes where it's all mostly driven by one eye and stripes where it's mostly driven by another eye. Or here, what we just looked at, if you look at the orientation of bars and you plot here which orientation of the bar, the color code represents which orientation of the bar most effectively drives neurons in these different locations across the cortical surface, then you see a regular arrangement of that nature shown here. So, or neighboring cells tend to have neighboring preferred orientation, and you get a 3D map. The 2D retinal surface is on top of this, and then the third dimension is the orientation. Of course, it cannot be a totally continuous map because 3 dimensions don't fit into 2, so you have to have sort of piecewise representation of cortical surface, and you can do this for color and many other dimensions. So from this comes the notion of a feature map, of mapping feature values like that about the stimulus onto the cortical surface. Now, in these kind of representations, you're characterizing a particular location on the cortical surface by the preferred value of the feature dimension, like the center of the receptive field and the preferred orientation. So, for instance, here, if this is tuning curve for orientation, represents the stimulus optimally, then this would be the most activated neuron. But you, of course, also have these tuning curves can be quite broad. You also have that the neuron might be firing for orientations that are not optimal. So the notion of population representation is that these other neurons also contribute in the form of averaging. It would be some kind of, if it's an angle, a vector averaging. And that is the predominant interpretation that we're using these days. It's not some kind of just reading of the maximal location, the maximally activated location, but that the entire population represents, contributes. And there is empirical data that supports that. Historically, perhaps the first study of that nature was in colliculus superior, so not actually cortex, a subcortical structure in David Sparks' group, where you also have a topographic mapping here of saccadic endpoints. So if you make a saccade into different directions, then a population here will become active. For instance, a population around A will become active when you make this kind of saccade, and B will become active, make that kind of saccade. D will be active if you make that kind of saccade. You can actually also conversely electrically stimulate a population here. It's actually different layers, but you can manage that and then elicit a saccade in that direction. So there's a very good mapping, tuning between saccadic endpoint and the location here in this topographic map. Now, that topography was used by the group to deactivate neurons in a targeted way. They could do that, for instance, by cooling the neural population for a certain amount of time, by bringing in an electrode that has some cooling mechanism. And by cooling the cells, they could turn them off. So, for instance, if this dark area is an area in which neurons are turned off, then while that's the case, they found that a saccade that is visually elicited to be in this B direction, having a visual target there that would be usually supported by that, was then systematically deviating from B. So the monkey was making sort of off target. And that would be explained if all the neurons here in this area around B that are typically active were contributing, and now because there's some contributing to the final saccadic endpoint, and now because some of these neurons here are not active, the center of gravity here of this population is shifted away from B in this off direction. So there are many other wrinkles of this experiment, but this would be proof that even these neurons around A that are sort of on the periphery of the population responsible for B do contribute. And there are very similar results like that in, for instance, area MT in the visual system and many others. So that's the background. This is tuning neural maps, and then the top population idea that the whole population contributes. So in neurofield theory, dynamic field theory, we're using these neural fields as mathematical descriptions of representations over metric spaces that reflect feature dimensions. And the difference is we're not thinking of these fields as being defined over the cortical surface. We're thinking of them more abstractly as defined over the feature dimension itself. And one dimension of one of those dimensions could be the retinal space, and that would be very much because of topography would be analogous or continuous with the cortical surface, but we're sort of abstracting away from the cortical surface and just mapping it into the original space. And I'll give you two examples of how one can do that operationally, and that will give you one particular wrinkle that we still need. So the first example goes back to work that I did with Janke and many other colleagues actually here in Bochum many years ago. And in that work, we looked at the anesthetized cat. AR17 is the primary visual cortex in that animal. And we did the conventional retinal tuning. So when we recorded from cells, we found out the receptive fields by showing sort of patterns of light projecting that onto their eye. And we found out the tuning to retinal location, the tuning curve. And the idea is that we then construct from a lot of these recordings a distribution of population activation by weighting that tuning curve, this sort of 2D tuning curve, with the activation of that neuron in any given condition. So for instance, in the conditions we then studied, we had particular stimuli, localized little patches of light across different retinal locations that were presented to all cells, right? The receptive field was found out individual cell by cell, but then we presented these stimuli to all cells, not across different trials. And then based on that procedure, you can directly estimate the population representation of retinal location. So here, this larger grid here indicates where this is on the visual array, and this little square here in the middle, that's where the stimulus was in these different conditions. And you will see that the color code is the distribution of population activation that now represents the stimuli. And you can see that it is sort of indicative of where that stimulus is. And so this is not on the cortical surface, this is directly on stimulus space, on the retinal space. And if you do that, you can ask interesting questions, for instance, about time courses. And here I just want to highlight one aspect in which we had composite stimuli that had two locations. Here, these two locations of brightness that were presented. And so we're comparing here on top the response to this composite stimulus with on the bottom the superposition of the individual responses to just the left or just the right stimulus. Remember, the left is closer to the nasal area, sort of closer to the fovea. And so they have more activity on the left than on the right. But the one interesting signature here is that you see that the response to this composite stimulus here is overall reduced over the superposition of the individual responses. Compare here on the right? This is a reduction, and that reduction is ultimately a signature of inhibitory interaction between those two populations. It's hard to see when they're close because you could wonder if this is just because there's some kind of rate limit. But here, when they are not overlapping this individual distribution, you can clearly see that this is reduced in the presence of the other stimulus, even though the individual populations don't overlap. And that's, of course, what we posit in dynamic field theory, that there will be inhibitory interactions over large enough distances. If you make a more refined analysis, you find also evidence for local excitation. So give that in the longer form of the lecture. This is not just about retinal position, which is very close to the topography. But now if you look at orientation, as we have seen, orientation is sort of distributed more in a more complex way across the cortical surface. But now if you're looking at the tuning to orientation and the tuning to visual position, you can kind of combine these two into two-dimensional representation where orientation and spatial position are the two dimensions. And this is from Dirk Janke's work in the year 2000. There's a little bar moving to the right and a little square moving to the right. As it moves to the right, it has a defined orientation that's horizontal. And you see, if you look at different time slices, you see that the population distribution of activation is initially sort of flat, and then it develops a peak around the horizontal dimension. So it extracts this orientation that comes from the movement. And if you look at the spatial location that's encoded in the population, you see that it is moving with the stimulus. It's lagging behind the stimulus, but as the stimulus moves to the right, the peak here moves to the right. And if you combine these two into a 2D representation, then you see that initially the location is sort of marked here by this vertical bar, and it takes some time for the orientation to come out. So after enough time, you get this localized activity that the spatial aspect of which is indicative of the spatial location of the stimulus with, again, a delay. And the location along the second axis is the orientation of that bar, the movement defines. And just to show that this would be now the direct representation over stimulus space, location, orientation, abstracting from retinal location in the DPA, distributional population activation. Second example from the motor domain, this goes back to work that I did with Alex Ariles' team. This is Annette Bastian's thesis work a long time ago in Marseille. This is the Jacopoulos type of paradigm where monkeys make center-out movements. For instance, it starts from an initial starting position. The monkey actually gets some prior information about which targets will come up, and then a go signal, the target changes color, indicates the monkey to make that movement. The monkey has learned to make the movement only when that go signal comes. And you can now similarly construct from the tuning curves of these neurons, more firing for certain directions than for other directions. A population representation is really based on this idea that you take the initial tuning curves and multiply them with the current firing rate in any particular direction. So for instance, if that's the direction you're currently specifying, you have that kind of firing rate, and then you would get a population distribution that is the representation of this parameter in the monkey's motor area. So here an example from constructing that from about 100 neurons. Here the monkey gets the first cue which movement is going to be. You see already a little peak at the right movement direction, and then close to the moment when the monkey is allowed to move, this peak grows in size and is centered exactly on the right movement. And here's an example comparing this to a case where the prior information is more broadly distributed. See a broader distribution, it takes a little longer to reach the same level of threshold. And that is exactly what we then model in dynamic field theory, right? The notion of a neural field over movement direction that we saw in previous lectures is meant to account exactly for these kind of patterns observed directly neurally. So here you see the direct match between neural fields and these distributions of population activation that are constructed from the response of many neurons interpreted in the space of varied external conditions, task or stimulus. One important aspect that I actually was driving at here is that in these pictures, the location in the field or in the DPA is not a neuron. The neurons are sort of spread out over the field, right? And this by construction here, and that each neuron contributes its entire tuning curve, and previously it was its entire receptive field that it was contributing, and therefore the neuron is smeared out over the field, right? So when we're looking at the field, loosely we sometimes said a field location is like a neuron, but that's not really true. It's a bit more abstract picture that at any location in the field, it will be multiple different neurons that are contributing to activation at that location, all those whose receptive fields or tuning curves overlap at that location. And so see the field abstracts not only from the cortical surface, but also from the sampling by discrete neurons. And that's also an interesting point of view. That's why we get these smooth patterns. And this whole idea of having continuity is a reflection of the continuity of the tuning curves and not a reflection of the discreteness of neurons, interior neurons. So in DFT, that's our units of representation. We're using these fields in different contexts, and from earlier lectures that the theoretical account for how the tuning comes about that then is constitutive of the fields is, of course, the forward connectivity, either from the sensory surfaces to the fields in question. So that will be the feature extraction, and there's a lot known about the neurocircuitry that gives rise to that. Or in the motor domain, much more complex, the projection from the motor field, like motor cortex, ultimately to all the muscles that then will make that certain kind of movement states arise. And I mentioned in an earlier lecture that involves really dynamics. That is, some motor state has a dynamical system. It's ultimately through reflex loops. The outer system is a dynamic system that has a loop state and loop and so on. We saw that in other lectures. So that's all I want to say at this very brief, in this very brief survey over the neural grounding of DFT. Thank you.